Hello students, welcome to EPG Pathshala. Myself Dr. Ashish Pal, Scientist E at Institute of Nanoscience and Technology, Mohali, Punjab. Today I am going to discuss the module Cooperativity, Synchronizing Noncovalent Interactions. It is under the paper Nanotechnology. The objective of the module is to understand how all these non-covalent interactions they act in concert, how you can actually synchronize all these non-covalent interactions, so that because non-covalent interactions are weak in nature, if they can all come together, then it can actually can be a force for reckoning. First of all, we would like to look at what are the different types of cooperativity effect present in synchronizing such kind of non-covalent interactions. We look into allosteric interactions, we look into chelate cooperativity, and when we look into chelate cooperativity and allosteric cooperativity, we would go into deep and try to look at the Hill coefficient, Hill plot, and interaction parameters from that. Secondly, what we would like to understand is how there can be flexibility versus rigidity between the molecules which can be governed by chelate cooperativity. Then how polyvalent interaction, the multivalency between two species, can be governed by chelate cooperativity. And lastly, towards the end, we would like to look at in macrocycles or macro bicycles how the effect of pre-organization works, so that this entire effect of cooperativity comes into picture. So this is overall the objective of the module as we go along. When we talk about non-covalent interaction acting in concert, the first picture of the story which comes into our mind is the Gulliver-Lilliput story, where one particular Lilliput may be very insignificant for the Gulliver, but when all the Lilliput they walk together, they can actually immobilize Gulliver on floor. So each non-covalent interaction is important, because even if they are weak, they can all come together and they can actually act together to get some important phenomena like protein folding or many other physiological phenomena. In that regard, if we look into a particular protein called streptavidin, which actually can bind very strongly with biotin, which is nothing but a vitamin H, you will see the binding constant of biotin streptavidin is very high. And if you look at the active site where the biotin binds streptavidin, you will see there are a number of hydrogen bonding interactions, cation pi interactions, and electrostatic interactions which actually takes place. So each of the non-covalent interaction, they contribute towards the binding of biotin and streptavidin. Cooperativity is the central concept to understand the molecular recognition and supramolecular self-assembly. Two classic examples of cooperativity is hemoglobin binding oxygen and protein folding phenomena. We know that when hemoglobin binds to oxygen, there is one unbound state, and after that, now hemoglobin has 4 sites, so oxygen can bind to all the 4 sites, that is completely bound state, but it goes to some partially bound intermediates. Similarly, in the protein folding, you have an unfolded protein and it goes to a completely folded conformation by some partially folded intermediate. Also, we can have supramolecular self-assembly where cooperativity is quite prevalent, where you may have an unbound state and you may have an assembled state where all the ligand and receptor they are bound to each other, but it may go through some partially assembled intermediate. So what is the signature of cooperativity? If we are talking about positive cooperativity, we can always have a look at the speciation profile, and we see in the speciation profile the change from unbound state to bound state is quite steep and it is sigmoidal in nature, whereas if it is a positive cooperativity, you see the accumulation of intermediate concentration has always been very low. So when we look at the binding isotherm of hemoglobin binding oxygen, and where we vary the pressure of oxygen, you see, and we plot fraction of oxygen bound to hemoglobin, we see it is a sigmoidal curve, along with that the transition is very sharp. And even in protein denaturation, when we take a denaturant and we change the concentration of that, we do see similar sharp transition behavior. So we can conclude, if the main signature of cooperativity is that it can have all or nothing, means all the molecules should be in bound state or none of the molecules in bound state. Let us look at the following scenario to understand cooperativity in much better way. The first system, as we see on your screen, that is the reference system, where you have two molecules of B and they are competing for two molecules of A. Here A is the receptor and B is the ligand, and they are binding to form this non-covalent complex AB. In the second scenario, we have one receptor where you have two binding sites, and that is where two ligand, monovalent ligand, they are getting connected and they are forming the bound state. Case number C, we see that there is a molecule where you can have both receptor and a ligand in the same molecule. That is why it is called AB. And at a particular concentration, these AB molecule, they can form intermolecular non-covalent complex, and it can actually eventually gives rise to supramolecular polymerization. However, in this case, there can be also cyclization when the complementary ligand and receptor, they can bind together each other one to one, and then it forms cyclized product. Now in the case D, where we have the receptor having two binding site, and even ligand is also bound together to form the same ligand is having two donating site ligand, and that can form ideally a cyclized product. To some condition, it may also give some polymerized product. Those scenarios can be understood in a better way when we take up an example. So here we are trying to understand what is allosteric cooperativity. Allosteric cooperativity refers to the phenomena where you have two binding sites in a molecule, and these two binding sites, they kind of influence each other. So let's say if you take the molecule which is on your screen, and the molecule binds to sodium. Sodium is actually the gray circle. It binds, and the molecule actually have a crown ether like complex. You also have something nitrogen which can actually donate to soft ruthenium. So you see, in one case, that is the left side of the molecule, you see that first the gray circle, sodium plus, is attaching and forming a crown ether non-covalent complex, and then in the next step the ruthenium is binding to those nitrogen and forming completely bound state. Now we can also have the other scenario where ruthenium is added first and ruthenium is getting bound to those nitrogen, and then followed by addition of sodium, which actually then binds to the crown ether complex. And in both the scenario, we can see that if you don't add the sodium or you don't add the ruthenium, then the corresponding association constant is pretty low when it is with the sodium or ruthenium. So we see in both the cases, when we add the sodium or ruthenium, the enhancement is more than around 40 times. So this is what is cooperativity, where the presence of another molecule, one binding site, that is actually influencing the other binding site. Let's try to understand this allosteric cooperativity with the help of binding constant and equation. The top system is our reference system. When A or B are joining to form this non-covalent complex, the association constant can be given by the ratio of the concentration of covalent complex divided by the equilibrium concentration of A and B. However, when we take this consecutive reaction where AA having two binding site is binding and forming non-covalent complex with 2B, in the first step you see that 1B is binding to form this intermediate, which is red color, AAB, and then other B is actually waiting to get bound to. And then in the first step we see that the association constant is 2K1, whereas this 2 comes from the degeneration of the state because you have two binding sites available. And then in the next step, the other B molecule that binds to the other binding site, and it forms this blue final product where it is completely bound state with a association constant half K2. Again, half is the degeneration factor, and the corresponding association constant can be given in the given equation. Now, if we take the ratio of K2 divided by K1, that is given by a parameter called alpha. This alpha is called interaction parameter. Now, this value of alpha, whether the system is cooperative or not. So, if alpha equals to 1, then it is independent binding sites. That means it has no cooperativity. It's just like the reference system. But if the alpha is less than 1, that means it is negative cooperativity. That means the first step, the association constant is so high that it is actually stable in the intermediate. Red complex is more stable than the blue complex. So here you can say that this is like an inhibitor. If alpha is more than 1, that means that is the case we are looking at more closely, and that is positive cooperativity. And next is how we quantify this allosteric cooperativity. So for we also introduce a term called binding site occupancy, which is equals to the fraction of receptor sites that are actually bound. So that is given by theta A. That means fraction of receptor site of A is bound, and that you can see that is given by total concentration of receptor site that are bound divided by the total concentration of receptor site which is available in the beginning. So now let's look at the consequence of this allosteric cooperativity. So for that we must look at the speciation curve, which we have already introduced a couple of slides earlier. So you can have the following scenario. So if you look at the first curve, where if I introduce you all the different stresses, so the blue trace, what you see is for the final completely bound product. The red one shows the concentration of the intermediate, and theta A, that is given by the black line, that shows the binding site occupancy. Whereas there are these gray dots, these are the one site reference receptors, means that is a reference system we talk about. Now when alpha equals to 1, it's just like the monovalent. So basically you always have this black trace and the gray dots superimposing on each other, and eventually you have a significant amount of intermediate concentration development, and then eventually you go to the final product, where if you plot these parameters with the ligand concentration. In the second case, where you have negative cooperativity, you do see a gradual response. So what happens is that initially your binding site gets occupied faster than the reference system, which is the gray dot as we see, but eventually that complex is quite stable, and after that the binding site occupancy becomes gradual with changing the ligand concentration, and we see there is a significant population of the intermediate state. And the last case, which is what we are all interested, is alpha equals to pretty high, something like 100. So this is where we see all or nothing behavior, because we see all the transitions from your binding site occupancy and the blue line, that becomes very steep, and there is no significant population of intermediate in this step. In a macroscopic scale, when we want to quantify this allosteric cooperativity, we must know what is Hill coefficient. So Hill coefficient is normally given by eta h, and that we get when we plot log theta a divided by 1 minus theta a versus the log of k dash b, which is the ligand concentration. This plot is called Hill plot, and once we plot this and we find out the slope of the origin actually gives the Hill coefficient, and you see the blue trace where eta h is more than 1, so that is what is positive cooperativity, and eta h equals to 1 means it's no cooperativity. It's like the reference system, and eta h less than 1, that is negative cooperativity. And if we look at the system where oxygen binds to the hemoglobin, as we know, this is what you see is hemoglobin's quaternary structure, where you have 4 subunits of hemoglobin, and Each subunit can bind to one oxygen and bind. Now what happens when it binds to one particular oxygen, then it has a positive cooperativity, and all the 4 binding sites of the hemoglobin gets populated by oxygen. And that's what you see in the saturation versus oxygen pressure, that curve. Do see it's a quite steep transition, and eta H is equals to 2.8, which is a positive cooperativity. It shows the all or nothing behavior. So let's understand another type of cooperativity, which is called chelate cooperativity. This cooperativity is little more complex than allosteric cooperativity. Here we see that nickel 2 plus get bound to 2 ammonia molecule with an 1 ammonia molecule to begin with an association constant K1, and then it binds to the second ammonia molecule to give this product. And as we know, for the consecutive reaction, the binding constant can be given, the overall binding constant can be the multiplication of the each binding constant. So that K1 into K2, that is 10 to the power 5 mole inverse to the power second. However, if we take 2 amino group connected by ethane moiety, that is a diaminoethane, then we see the binding constant, that is actually 2 into 10 to the power minus 7, in that order. So 2 order of magnitude higher. So then we can define a term called effective molarity, which is a measure of cooperativity. When we take this ratio of both the systems, so we see that value is around 400 molar. So the effective molarity is the measure of cooperativity, and it actually is very important to predict in supramolecular system whether there is an effect of chelate cooperativity or not. When we look into the chelate cooperativity in terms of equation, where we have one receptor with 2 binding sites AA, and there is one ligand with 2 donating site BB, and then they form this 1:1 complex of intermediates, which is an open chain analog, and then with a association constant 4K, where 4 is the degeneracy factor, and then this intermediate can either go into the cyclic, that is the green product, where it actually closes, and or it can also form 2:1 complex, which is also open chain analog, which is given in blue. And here the effective molarity you can define, which is equals to half KEM equals to the ratio of the closed product divided by the open chain product. And here you can define the cooperativity, that it is the concentration beyond which the cyclic structure they actually begin to convert to the polymer. When we look into the speciation curve for the system which has the potential chelate cooperativity, we do see in the first case where the KEM is lower than 1, that is 0.01, we do see there is a significant population of the intermediate, which is the red product, which is open chain analog, and there is development of this blue trace, that is the supramolecular polymerized product. But also we have the cyclic compound which doesn't really form at all. However, when we have KEM which is more than 1, that is around 100 in this case, we do see that there is a significant population of this cyclic product. However, beyond particular concentration of the ligand, the cyclic structure of the cyclic product, they get converted into this 2:1 complex of, it is like the open chain polymerized product. So this is very important, and we do see in this case of positive cooperativity, for chelate cooperativity, there is no development of concentration of the red species, which is intermediate. This is very important to know when you are making some nanostructure which has a potential based on the non-covalent interaction, which can potentially form cyclic analog or open chain polymerized product. You need to optimize the concentration of the reaction so that you get the desired product. So one direct consequence of this chelate cooperativity is multivalency, which is nothing but the enhancement of the affinity through simultaneous presence of multiple non-covalent interactions. So in this case, we see that this system has multiple receptor and ligand in the same system, and then they can cooperatively form non-covalent structure. And this is what we see when we look into the example of influenza virus, which is binding to the cell surface. As we know, influenza virus has hemagglutinin at their surface, and on the cell surface there is sialic acid, different forms of sialic acid, and they bind together. And so if you have to prevent, as you see from the cartoon image, if you have to really prevent influenza virus from attaching to the cell surface, you must add a similar polyvalent molecule which will actually prevent the binding. Let's look at this following example where a metal ion like potassium is binding to these different molecules through ion dipole interactions. So in one case you have this podant where it is binding to this potassium and it actually forms this macrocyclic like structure. Another case you have something called corant, it already have this macrocyclic structure and then it just binds to the potassium ion. And in the third case you have a macro bicycle which is called as cryptant, and it encapsulates the potassium over here. If you look at this three system, if you look at also their association constant, you see as you go from this podant to corant and cryptant, the association constant increases. That means the complex become more and more stable. So this phenomena is known as pre-organization. So if you look at, in the case of cryptant, there is already macro bicycle is organized, and what you have is lone pair repulsive interactions, and these lone pairs are not solvated and there is little change in terms of enthalpy. But however, when you look at macrocycle like crown ether, it is somewhat less lone pair impulsive interaction and somewhat solvated. But if you look at this podant, there, even though there is little repulsion of lone pair, but they are very highly solvated. And when you get them into the macrocycle, you see that there is the solvation effect plays a role, and because there is a high enthalpy change which is involved in this case. So if you look at the following example where you have a macrocycle and in other case you don't have a macrocycle, it binds to copper or zinc and it forms a macrocycle like template, you can see in the first case the enthalpic change is much more than in the second case, and this has to do with the pre-organization because of this lone pair being solvated, and then once you get them back into packed together, then first of all they have a charge repulsion and you also sacrifice the solvation. So let's summarize the module, what we have learned so far in this module. So first of all, we learned how different non-covalent interactions they act together, and then they become a force in reckoning in stabilizing a particular complex. Now in that regard, we have looked into two different types of cooperativity. One is allosteric cooperativity, one is chelate cooperativity, and we have actually compared and we have seen, with respect to the reference system. Now if you change and the number of binding sites available, you can actually get into completely different scenario. So allosteric cooperativity is much more simpler, and we can see the effect of cooperativity and how the speciation curve or the change depending on the alpha value, that if it is more or less, or if it just behaves like if there is no cooperativity. So all these we have learned, and this is important when we look into some journal articles and all. This will be really helpful in understanding the experiment which a scientist they perform. Secondly, we have also learned how to plot the cooperativity and quantify the cooperativity using Hill coefficient. For that, we can plot in Hill plot, and we look into the slope at the origin, and we can figure it out depending on the slope magnitude. We can figure it out whether it's a positive cooperativity, negative cooperativity, or it is no cooperativity at all. Then we slowly moved into little bit complex kind of cooperativity, that is chelat cooperativity, where unlike allosteric cooperativity, you have two binding sites in a molecule, and also you have two ligands which are joined together, and it can form then complex, which can be first of all, it can be a cyclic complex, or it can also form 2:1 complex like that, and it can actually form supramolecular polymerization. But that actually will be governed by something called effective molarity, and that it all depends on the concentration under which you are performing the reaction or binding study, because based on that concentration, you can have either cyclic structure, or if you have more, then you can have intermolecular binding, and that will actually give you supramolecular polymerization. So this idea of chelat cooperativity can actually be very useful later when you actually do some experiment and conduct a study, and if you are specifically looking for one of the products, let's say you are looking for cyclic products, then you know that in case you have to do this intramolecular cyclization, then the corresponding uh, concentration regime should be much lower. So, and that also depends on the K effective molarity of the system. Now having done that, we also looked into different types of like thermodynamics, pre-organization, multivalency of this kind of chelat cooperativity. We have seen that uh, if the host, where let's say the guest is binding, host is already pre-organized, then it doesn't have to really pay that much of enthalpic penalty as it is when it is in open chain, because then in open chain there is the solvation which act against the folding of the let's say the podant to form the inclusion complex. So we have looked into the pre-organization, the thermodynamics. We have also looked into the multivalency effect, that is also a part of cooperativity, and how we can really specifically bind using a the polyvalent ions if it is ionic interaction, and we can exclusively have a complex. So I hope this module is very helpful for you, so that you can study further many research papers and you can actually plan your experiment accordingly. Thank you.